Hey Beaklings, today we're going to wrap up Unit 8, which was mitosis, the cell cycle, cell division, and meiosis. Okay, and what we're going to talk about is why meiosis leads to genetic diversity in our sexually reproducing population, and we're going to talk about some bad things that can happen if meiosis goes wrong. Okay, now you shouldn't really stick with me on this video yet unless you really understand meiosis. So if you need to go back over the ins and outs of the process of meiosis, go ahead back and redraw, watch my other videos, and then come back to this one where we could really dive in. Okay, so here we go. There is some similarities and some differences between mitosis and meiosis, right? So they're both forms of eukaryotic cell division, right? But the purpose of mitosis, or division of the nucleus, and then eventually the cell via cytokinesis, right, is growth, tissue repair, asexual reproduction, but you're going to make two genetically identical daughter cells. That's important, right? So these are those somatic cells. Whereas meiosis, no, that's where you create four dissimilar haploid gametes, right? You know, gametes are our sex cells, okay? So now it's important to remember you do not go back into the cell cycle here, right? When you're at finishing up meiosis 1, telophase 1, you go right into prophase 2. Okay, so the chromosomes did, uncoil back into chromatin. They're going to coil right back up for prophase 2. So you do not go back into interphase. That's important because you'd be recopying your chromosomes, right? You just made haploids. Why would you want to go back into S phase and make copies again? So mitosis only had one nuclear and cytoplasmic division, whereas meiosis, it happens twice, okay, for each. And all unique events to meiosis happen during meiosis 1, because meiosis 2, if you remember, that's really just mitosis with half as many chromosomes. So meiosis 1 is where the magic happens, okay? So let's take a look. What do you need? Okay, so let's take a look here at what we have. So the two major things that happen in meiosis that lead to genetic diversity happen in meiosis 1, prophase 1, and metaphase 1. So what is it about prophase 1 that's so important? Well, you remember synapses, right? We pair up homologous chromosomes. So number 1 from my mom and number 1 from my dad come together, okay? And if you notice here, this synapsis, okay, is where the crossing over will happen. So here you can see 1, 2, 3, 4 sister chromatids. That's why it's called a tetrad. And when they cross over, no big deal, right? Because if you think about it, what are you actually crossing there? You're crossing, nucleotide sequences. But if you're crossing genes, well, you're crossing the same gene with maybe different alleles. Okay, so you're still getting all the right genes, but maybe you're just getting a different variant of that gene. So crossing over is really cool, and it leads to diversity of our species. You're basically shuffling your genes by doing that. And guys, you're doing it right now while you're sitting there, and girls, you've already crossed over your eggs before you were even born. And in metaphase 1 now this one's a little trickier. Students have a hard time wrapping their head around metaphase 1 and why it's so important. And it's because of the orientation of the chromosomes, right? These chromosomes align themselves independently, and then, they eventually will get separated and based on how you lined them up, right? This independent orientation or independent assortment of your chromosomes is really, really, really hugely important as you get into some advanced genetics. Okay, so let's take a look here. In other words, in metaphase 1, we have only 2 pairs of chromosomes in this cell. Obviously, you have 23 pair in your cell. Okay, so here, let's just say this is mom, and then this one is dad. Well, then maybe this one is dad, and this one is mom. Oh, there's all different ways to do it. Or maybe you went mom, dad, or maybe you went dad, mom, mom, dad, or dad, mom, right? There's all different ways that you could set up these chromosomes. So, and what the equation is 2 to the haploid number. So if you think about a human being, that's 2 to the 23rd power. That's over 8 million possible combinations. So just think about that. When you make or made your sex cell, that means that you had basically a 1 in over 8 million shot of setting it up in a certain way. That's a huge amount of diversity, right? Because you're mixing up all your way that you're setting up all your different sex cells. That's not even mentioning crossing over, by the way. That's just how you set them up. That's crazy. Okay, so metaphase 1, very, very, very important. All right. And what about anaphase 1? All right, well, this one is not as important for diversity, but it's important to understand the difference. Look what you did here. You did not separate your sister chromatids, right? What you did was you separated this from this, right? One chromosome goes this way, gets pulled, the other chromosome goes this way and gets pulled. You never actually separated sister chromatids at the centromere, right? So you still have full-fledged chromosomes. And that's why you're still going to have 23 by the time you're done meiosis 1. That's how you get a haploid, because you took 23 and went this way and 23 and went this way. A lot of kids don't understand why at the end of meiosis 1, you're haploids. You're not usable yet as a sex cell, but technically you only have 23 chromosomes. Okay, and that's because in anaphase 1, you separated the homologous pairs from each other. Okay, so comprende. Explain why mitosis conserves chromosome number while meiosis reduces the number from diploid to haploid. Well, of course, mitosis, again, you have a single replication and one division. So you copied in S phase your chromosomes and then you rip them in half, good to go. However, meiosis, you still just have a single chromosome replication, but then you divide first time and then you divide a second time. Oh, that's how you end up with half as many, right? Okay, here we go. So I often tell students, if you're a little confused on mitosis versus meiosis, this is a great picture. And there's one just like this online, and maybe I gave you something similar to this in class. But this really gets you to wrap your head around it, because look at what it starts with here. This is just your cell. Now again, you would be a 2n equals 46 cell, but this is just a 2n equals 4 cell. And look at the difference here. In mitosis, sure, you coil up your chromosomes, you line them up, and you rip them in half. Look what you ended with here. 2 genetically identical daughter cells, and look who they're also identical with, the cell that you started with. They're identical to the parent, right? So maybe this is like a G1 cell, although they wouldn't be coiled up there. Now, look at the difference over here. Oh, huge difference, right? Look at meiosis 1. These crazy things that happen in meiosis 1 are much different than over here. And when you're all done, take a look at these cells. They're all going to be genetically different from each other, and look how different they are from the original parent cell, right? So meiosis, mitosis, very, very different. Okay, so what we're going to do now is we're going to go right back into it here, and we're going to talk about, well, how independent orientation of chromosomes in meiosis and random fertilization lead to varied offspring. Okay, now you are hopefully somewhat aware that we talked about last time, mutations are the key. They're the backbone of genetic change in basically any species, right? This is what makes evolution make sense. Okay, and evolution is the backbone of biology, and it's driven by these random mutations that can happen. All right, so if you want to learn more about that, we don't cover too much deeply in Bio 1, okay, but go on to Bio 2 Honors, AP Bio, another bio class someday, and you're going to dive way deeper into population genetics. Okay, so what we're going to talk about here is not necessarily mutations, but how else are we so different? Well, thanks to sexual reproduction, whether or not you get a certain gene from your mom or dad is as random as flipping a coin, right? It's 50-50. So we're actually starting some genetics thinking right now. And remember that even though homologs have the same genes, they very well might have different copies of those genes. Okay, so if you remember back, you can see these lovely ears right here. So if you remember, my dad had big, huge, gorgeous, handsome, yeah, I'll say it, sexy earlobes like this, whereas my mom has attached earlobes. So what? That makes me a carrier. That means I have a gene or an allele for the free earlobes, and I also carry inside of me, you wouldn't know it, an allele for the attached earlobes. Okay, it doesn't matter. I still have 2 copies of that gene that I could randomly shuffle and line up however I want and give to my kid. Okay, so that is homologs having different copies of, you know, the same genes or different alleles perhaps. And some of you out there might have the same allele, which we'll talk about. So the independent orientation thing, this is kind of weird for, like I said, for some kids. It just means that you could set them up however you want in metaphase 1. So here you can see 2 possibilities. Just looking at this versus this. We're not even talking about crossing over or anything like that. If you set it up dad, mom, look at what your sperm or your eggs would have looked like. Whereas what if you just change one thing here, dad, mom, and oh, mom, dad. Well, now look at this. Whoa, very different from each other. Okay? And like I said, you have 2 to the nth power. You have 2 to the 23rd power, over 8 million possible combinations for you to, you know, create different gametes. Okay? That's pretty impressive stuff right there. So part of the reason that we're so diverse as a species is because even just one individual organism, a member of our species, look at the amount of different sperm or eggs that you can make just based on how you line them up in metaphase 1. Not even talking about shuffling or anything like that, just how you line them up. That's crazy. Okay? So now let's flip the coin here. Oh, Well, what about it takes two, right? I just told you about if you make your sperm or eggs, that is about a 1 in 8 million plus chance of making a very specific sperm or egg. That doesn't create an offspring. It takes two, right, to tango. You need two organisms coming together for sexual reproduction. So think about that. That's what random fertilization is all about. How do you know what sperm is going to meet with what egg? And don't forget, this person who made this sperm had a 1 in, over 8 million shot. Let's just call it 8 million. A 1 in 8 million shot of making this sperm. And this person had a 1 in 8 million shot of making this egg. And for those two randomly created cells to meet and fertilize to form a zygote, a diploid, whoa, that's about a 1 in 8 million times 1 in 8 million, 1 in about 64 trillion chance. More than that. That's random fertilization. So because of you lining them up in metaphase 1 in different ways, you have about a 1 in 64 trillion chance of the exact, same sperm or egg coming together based on how you made them. That's crazy. And what a crazy way that our species is so diverse from each other. And by the way, that's if, this person right here and this person right here had a kid. Okay? But what if you have a brother or sister who's maybe not from the same two parents that you had, right? The same two biological parents? Well, then it's even more diverse. Right? It's crazy. Our species is very, very healthy when you think about how jumbled up our genes are and things like that. And that's a good thing, right? Because the closer your genes are sometimes, the more likely that can lead to problems, which we'll talk about in genetics. Okay? So don't forget, though, that 64 trillion, just the beginning. Because what didn't I mention in that calculation was crossing over between homologous pairs. We can't even calculate that, right? At those chiasmata, right? You have 2 or 3 per chromosome pair, might be more than that even. And if you had to calculate, well, where am I going to cross and how are you going to cross and all that shuffling that goes on, that's astronomical, right? So it's crazy. So a lot of times I'll have kids in class ask me, well, what are the odds that the two people that made me could have had another person that's exactly the same as me genetically that's not an identical twin? It's astronomical, right? There's no possible way. It's crazy because of crossing over is the shuffling. So it wouldn't be 1 in 64 trillion. It would be 1 in 64 trillion times the exact same crossover events. That's crazy. Okay. All right. So comprende. One species of worm has a diploid number of 10. How many chromosomal combinations are possible for gametes formed by meiosis? So what you should be thinking right now is if the diploid number is 10, that means the haploid number is 5. And that means 2 to the 5th power. So 2 times 2 is 4, times 2 is 8, times 2 is 16, times 2 is, right, 32. Okay, so hopefully that one made sense to you. Okay, so now we understand independent orientation and hopefully random fertilization. Let's go back over crossing over again. And we've been hammering this home, so hopefully this is making sense. But homologous chromosomes carry different versions of genes, sometimes, like we said. So here you can see 2 mice, and you can see their homologous chromosomes. And it's because of these different versions are what makes gametes different a lot of times. So, for example, I can see here that there's a mouse that has a chromosome from his mom, maternal, and a paternal chromosome, and these are homologous. And why do I know they're homologous? Well, look at this. The coat color gene is right here on both of them at this locus. Okay, and then there's another locus here. So there's only 2 loci that we're looking at here. One is for the coat color and one is for eye color. And in this case, you can see dominant is brown and for coat color, and white is recessive. And I know that because of this capital and this lowercase letter, which we'll talk about more with genetics, the symbols that we use. And then black is the eye color, but yeah, maybe recessive here we'll say is pink. So if you look, this mouse right here would actually look just like him. Okay, even though inside he carries this, looks like his dad must have given him, these alleles, and his mom gave him these alleles. Okay, so same chromosomes, same genes, but different alleles in this case. Doesn't have to be different, but they are. Okay, so when you do cross over, that increases genetic variability. Okay, and it's again exchanging corresponding segments between homologous chromosomes. So here you can see your tetrads. Now we're early in prophase 1 here when you're going to cross over, so they're not tightly wound up yet. Okay, they're still pretty stringy, these chromosomes as they coil up. And if you remember how they coil, it's a very complex process. You can watch the other video on that. And chiasmata are now the sites where they're going to cross. Okay, and look at that. That is so random. We have no idea where they're actually crossing there, right? So definitely increases genetic variability. So let's go back to these mice again. Okay, so we said maybe this mouse right here, okay, maybe this is his chromosomes. Let's say that's the boy. And well, what's going to happen here? Okay, he can actually mix it up a little bit when he goes to make gametes. Okay, so here's chromosomes right here. Let's say that they're up here, and let's say that this is prophase 1. Well, if you notice, they're showing like a break in a crossover event happening right here. Just one crossover event, of which there'd be a lot more than just one, likely, right? And if it just crossed that one time, we'll take a look here. Okay, there's a different chromatid than existed earlier, and this is a different chromatid that existed earlier. The outside up here is still all maternal, and the outside down here is still all paternal, but the internal chromatids have now changed. And just based on one pair of chromosomes with one crossover event, look at what we got down here. 4 different chromosomes, right? Daughter chromosomes would end up in the haploid gamete. So just based on one crossover event between one pair, holy cow, all right? And hopefully now you can understand why crossing over, think of all the crossover events that happen for all 23 pair of your chromosomes, lots and lots of diversity, right? Okay. So crossing over produces genetic recombinations of genes that otherwise wouldn't exist, right? Because you're taking your mom's DNA that she gave you and your dad's DNA that he gave you, and you're randomly shuffling it together. That doesn't normally happen in nature. That took you making gametes in your sex organs to be able to do that, right? And again, we average 1 to 3 crossover events per chromosome pair during meiosis. So wow, your gametes probably aren't even representative of your chromosomes, right? You shuffled them all up and now they're all crazy different. So hopefully by this point, you feel pretty comfortable about 3 sources besides mutations, of course, 3 sources of genetic variation. Independent orientation, right? That's metaphase 1. Random fertilization is not really meiosis. That's when a random sperm and a random egg would come together, right? That's that 64 trillion chance there. And then crossing over, as you know. Okay, so here is a quick video on crossing over. Okay, and if you feel comfortable crossing over, you can go ahead and skip this almost 2-minute video and move along to the next section. But if you'd like a little refresher, here you go.
Crossing over during meiosis allows recombination of genes between homologous chromosomes. This alters the linkage between genes on the same chromosome. Crossing over occurs frequently during prophase 1 of meiosis, when homologous chromosomes associate with each other in a structure called a tetrad. First, consider the possible inheritance of alleles for 2 genes on a particular chromosome when crossing over does not occur. Recall that one member of each pair of homologous chromosomes came from one parent. The other member came from the other parent. In this example, one parental chromosome had alleles capital A and capital B, while the other had alleles lowercase a and lowercase b. Without crossing over, each haploid cell produced will inherit a chromosome with the same combination of alleles as was on one of the parental chromosomes. Thus, the arrangement of linked genes is not altered in this case. Now, consider the case where there is crossing over in the region between the 2 genes. Since the crossing over is only between 2 individual chromatids, the possible haploid cells after meiosis have 4 different genetic combinations. 2 will come from the chromatids that did not participate in crossing over and thus will still have the parental combination of alleles, either capital A and capital B, or lowercase a and lowercase b. The other 2 will be recombinant, with either capital A and lowercase b, or lowercase a and capital B alleles. Thus, crossing over allows for the reassortment of linked genes.
Okay, so hopefully that was helpful. Now we're going to talk about what happens when there are mistakes or accidents that happen. Okay, and nondisjunction is a very important vocabulary word. So in some rare cases, homologous pairs of meiosis 1 or sister chromatids, right, which would be meiosis 2, fail to separate correctly, and this results in abnormal numbers of chromosomes in gametes produced. Okay, now again, you have to be pretty sharp at meiosis to really wrap your head around this, but if you understand meiosis 1 and meiosis 2, you should be able to see why an event in meiosis 1, a nondisjunction event, would really affect all of the sex cells, whereas a nondisjunction event in meiosis 2 would only affect 2 of the sex cells. So let's take a look here. Okay, here you can see nondisjunction, fails to come apart correctly, right? And in this case, instead of the homologous pair separating in anaphase 1, whoa, they stayed together on this side, and now look what happened. Well, this cell in meiosis 2 now has an extra set of chromosomes, and this one is missing a chromosome. Oh boy. And look what that's going to mean. Now, all your gametes are going to be different. This gamete has an extra one. This gamete has an extra one. It's not a haploid. It's not N. N plus 1, N plus 1. This is missing. So N minus 1, N minus 1. Now, if the non-disjunction event happened in meiosis 2, okay, so everything in meiosis 1 looked good, but now in meiosis 2, you know that we separate the sister chromatids from each other at the centromere, creating daughter chromosomes. Well, look what happens here. If you pulled both sister chromatids to one side, oh, that's going to be problematic. Now, over here, we did it okay. So our haploid gametes look to be okay, right? We have 2 N cells here, okay? Over here, though, well, this cell now got 2 copies of this chromosome, so you're going to be N plus 1. And over here, we didn't get any. So when you split, we have N minus 1. So non-disjunction is actually a pretty big deal, and it's a little bit scary that this stuff can happen. Okay, and here you can see Down syndrome is a very famous disease, a well-known, well-studied disease that's caused by non-disjunction. And in this case, they're showing that, well, it was a result from an egg. And a lot of times it probably is due to the egg, more so than the sperm. We'll talk about why in a second. But, yep, normally you should have just 4 chromosomes, well, and you know you would have 23 and 23 to make 46. But in this case, instead of 4, we have 3 plus 2, so that ends up with 5. So here you have a diploid chromosome, but that extra chromosome in there causes a lot of complications. And a lot of kids often ask, well, what is going on there? Why would having just an extra part of a chromosome or an extra chromosome altogether, wouldn't that be a good thing, right? And no, your body is designed to have 46 chromosomes. So generally, if you goof that up, especially your autosomal chromosomes, it's not going to be a good thing. Okay, so let's see here. You can have a karyotype performed. And a karyotype basically can check for errors that occur during meiosis. Okay, it's a magnified picture of an organism's condensed chromosomes. And oftentimes what they want to do is get a picture of like white blood cells that are dividing. And then scientists can stain them and you get this beautiful picture here, right? And in this case, you could see this looks to be a healthy boy, right? XY in there, and you can see that they lined up all the chromosomes. Don't forget, this is not an X right here. This is actually these 2 chromosomes right here. This homologous pair is what's being shown right there in that box. So there's actually 4 chromatids right here. Okay, so this is all 46 of your chromosomes, all 23 pairs. All right. Whereas you might have a normal karyotype or you might have an abnormal. So again, here you can see a normal karyotype. Okay, and they're arranged longest to shortest, and then you have your sex chromosomes, which are a little bit different, right? Our sex determination system is decided by our sex chromosomes. They don't act exactly the same way as autosomes. They're a little different. So what can you tell me about this karyotype here? Okay, is this a dude or a dudette? Well, you can see right here it's an XY, so that's a dude, right? And is it healthy? Yeah, it looks like it. Okay, and I told you they're arranged, you know, longest to shortest. There is some controversy there, whereas number 21 actually does look to be a little shorter than 22. It's just when scientists first set them up, they thought this was correct, so they leave it that way. But it does look like maybe 22 should be over here, be number 21, and 21 should be over here as number 22, but it's not worth changing at this point. Okay, so they're both pretty similar and small. Okay, so an extra copy of 21 causes Down syndrome. Okay, so trisomy 21. A syndrome is when you have a characteristic set of symptoms. Okay, and we'll talk about what Down syndrome is in just a second. But it affects about 1 in every 700 U.S.-born infants, right? And it's our country's most serious birth defect. This is a big deal. So what you can see right here is that this is a girl based on this karyotype, right? XX, because if this is a Y chromosome, it would be itty-bitty, right? But these are 2 X chromosomes. And you can see right there that she has 3 number 21s. Okay, so she's experiencing trisomy 21, and because of that, she's going to end up with Down syndrome. Okay, so what is it about Down syndrome here? We said there's a characteristic set of symptoms, and I'm sure you either know people with Down syndrome or you've at least seen them walking around school or other places. They're a round face, skin fold at the inner corners of the eyes. A lot of people think they look very happy, right? They have a nice face and flattened nose bridge, small irregular teeth, very often a short stature. But there's some very serious things that go on here too, like heart defects, sterility, susceptibility to respiratory infections, leukemia, Alzheimer's disease, right? And all of this because of that one extra chromosome, which, like I just said, it's your smallest chromosome. So your tiniest little chromosome having an extra copy of that, that's trouble, right? So this little autosome in there, the very end of all your autosomes, can cause all of this, right? Your body's designed for 46. It's just how it is. It's a shame. So most people with Down syndrome have shorter than normal lifespans and have varying degrees of mental retardation. Now, some people have joined the workforce, and some there's even movie stars and actors and things like that. But generally, this is a pretty tough disease to deal with. Um, some women with Down syndrome, now we said they're sterile, but there have been cases where some women with Down syndrome were able to reproduce. Okay, and there's often some not so great stories that go along with how they got pregnant in the first place. But if you think about it, 50% of their gametes would be messed up, right? Would have that extra copy because they ended up with three number 21s. Think about what that means. If I have three 21s, well, even if I put one 21 on this side, sure, but that means I'm going to put two on this side. And that means that any gametes that come from that side are going to have that extra chromosome. So at least 50% of the gametes will have this trouble, okay, if somebody with Down syndrome would be able to reproduce. And as you can see here, the risk of having a child with Down syndrome increases with the age of the mother. Now, that doesn't mean that males can't have non-disjunction go on. No, no, no. It just means that it's very likely, well, maybe that's the wrong word. It's if there's a child born with Down syndrome, it's likely that it came from the woman simply because don't forget, you made your eggs while you were in your mom's womb. You just didn't finish meiosis quite yet. So now, when you're going through ovulation, you go ahead and finish that egg. And if you're talking about 40 plus years have passed, well, that's a long time. Whereas guys make sperm every day, all day, okay, for the duration of their lives. So it's a little different. So we don't know for sure, but scientists speculate that maybe it's just because these cells have been laying there dormant for so long, and the older you get, the more chances you have for a mistake. Okay, who knows? Here you can just see a comprende question. I don't know if I agree totally with the way that the graph is being read here, but this is from an old book that we use. So it says for mothers of age 47, the risk of having a child with Down syndrome is about how many? So as you can see, as the age goes up, so 47 would be right about here, right? So if you go up here, they're saying maybe it's about here, 40%. I don't know. I think it's almost up to 50 at that point. But if it was 40, that'd be about 4%, right? Because it's per 1,000 births. So the important fact is that, yeah, as the age of the mother goes up, the risk of Down syndrome certainly goes up as well. And that's pretty much all we talk about. There are other autosomal non-disjunction diseases, but we pretty much end our discussion right there because oftentimes if you have non-disjunction at an autosomal chromosome 1 through 22, you're not going to have a successful pregnancy or give a successful birth. It has happened, but a lot of times it's, yeah, we might get away with that extra 21 because it's so tiny. However, we don't get away with most of the other ones. It's almost like the mother's body maybe can just sense that something's not quite right developmentally, and there's a natural miscarriage. Okay, but having said that, there are other non-disjunctions that are actually somewhat common, and they happen at the sex chromosomes. They don't seem to affect us the same way as autosomes. So non-disjunction at a sex chromosome is a little different. Let's take a look here. So abnormal numbers of sex chromosomes do not usually affect survival. So here's two examples right here. Here you can see a male, and no matter what, you know it's a male, not just based on the anatomy, but you can see that, oh, there's a Y chromosome. As soon as you see there's a Y chromosome, you know that you're talking about a male, okay? And what happened here was, oh, the male through non-disjunction got an extra X chromosome. So instead of 47, or I'm sorry, 46 XY or XX, he happens to be 47 XXY. Okay, now how did this non-disjunction happen? Who knows? Maybe the mom gave him an extra X or the dad gave him an extra X, right? Instead of just a Y. Who knows? Okay, but point is, he's pretty much fine. You can see there's some characteristics here. Poor beard growth, maybe some breast development, and underdeveloped sex organs, sure. But a lot of times, guys might not even know that they had Klinefelters unless they were trying to maybe have a child because they are sterile. Okay, and maybe they couldn't have a baby and they went to a specialist and they did a karyotype and said, oh, guess what? You know, you might not have ever known this, but you actually have Klinefelter's disorder. All right. And similarly, now this is obviously a woman, and why do I know that this is a female? Okay, I should say and that's because it's no Y. right? If you don't see a Y, then you know you're not talking about a male, you're talking about a female. And in this case, there's a missing chromosome. Now again, did this X come from the mom or did it come from the dad? Well, it doesn't matter. You're missing the other one, right? There's only one sex chromosome in this case. And again, she's okay. She has some characteristic features, okay? Little web of skin here. Sometimes it has like a little bit of a squarish or a masculine look to it. Constriction of the aorta. Okay, well, that's not good. All right, so that can lead to some complications. Again, poor breast and underdeveloped ovaries as well. Poor breast development, poor ovary development. So again, this girl might be completely normal. She might have lived her whole life and not realized anything was different. And then maybe she was trying to have a kid someday and it wasn't working out. She went to a specialist and, oh, guess what? You're missing a chromosome. And we call that 45XO or Turner syndrome, right? So Klinefelter, you have an extra chromosome because of non-disjunction. And in this case, you're a guy. And in Turner syndrome, you're missing a sex chromosome because of non-disjunction. And in this case, you're a girl. But both of these can have very normal, regular lives. All right, and here you can see some other things going on. So here we already covered Klinefelter and Turner, but in this case, XXX. Okay, this is a normal female. Okay, actually that girl right there might not ever know that there was something wrong. So you can see, yeah, sex chromosomes don't act the same way because the X chromosome is actually very big. So the fact that you have an extra one and it doesn't even affect you, that's interesting. And in this case, XYY, yeah, normal male. So you can have an extra little itty bitty tiny Y chromosome and you still develop normally. There has been some studies about hormones and things like this. For example, maybe some extra testosterone or you know, maybe a little bit more muscle or violence in certain people, like aggression and things like that they've been studying. But for the most part, these are just very normal. Okay, just normal right here. Whereas this one is a disease, but you're still okay. And this one is a disease and you're still okay. So messing up the sex chromosomes through non-disjunction is not the end of the world. So we do have one last thing to talk about, and that is not missing or gaining a chromosome, but actually what happens if the chromosome itself gets a little messed up, which can also happen. Okay, so alterations of chromosome structure can actually cause birth defects, sometimes cancer as well. All right, and these I tell my students all the time, just don't worry too much about anything too deep yet until we get into genetics, but you can tell what's going on here just by looking at the picture, right? Deletion means you deleted a section of the chromosome. Duplication means that, oh, now you have a double section of the original one section, now you have two. And inversion simply means that you flip them, okay? And that some of these are very serious. For example, deletion, yeah, you don't want to be missing genes. You might say to yourself, hey, wait, but don't I have homologous chromosomes? Wouldn't I have the same genes on the other chromosome? Yeah, but again, your body's set up to be diploid, two good chromosomes. So even if you just mess up one, that could really goof things up. See an example in a second. And duplication, again, this has happened, and sometimes you get away with it. Sometimes it can be a little problematic. And even inversion. Your cells actually know where your loci are for different genes, and if you start mixing them up, that can lead to problems. Okay. So, one problem here, this cri du chat or cry of the cat syndrome, very, very, very sad. And what happens are these kids eventually die at young ages. And the only problem is on chromosome 5, if you look here, there's a deletion. They're just missing the end part of one of their chromosomes. And again, it kind of baffles scientists, and it's super sad because, I mean, they have one full good number 5. But because the other chromosome number 5, you have that deletion, it's pretty sad, and it's eventually a death sentence. Not good. And here you can see translocation can also happen, and this is another one where I like to, if you're good now with this stuff, I like to call this crossing over, but with non-homologous chromosomes, right? You all already know now that if number 1 from my mom and 1 from my dad cross over, that's okay. That's just going to increase genetic diversity, right? Like, for example, my earlobe attachment, it's free versus it's attached, depending on which parent chromosome I'm talking about inside of me. Well, who cares if they switch? But what if I changed on one chromosome for earlobe attachment gene? What if I cross that over for how I make my neurons in my brain? Oh, that's huge. You don't want to be crossing things like that over. That's what translocation is all about. Okay, non-homologous crossing over, that could be bad. All right, so let's take a look at a kind of a neat story here. Well, neat and a little sad as well, obviously. A chromosomal translocation in somatic bone marrow cells is associated with what we call chronic myelogenous leukemia. Okay, as the diagram shows, when chromosome 9 and 22 reciprocally translocate, meaning crossing over between non-homologous chromosomes, okay, a cancer-causing gene becomes activated in what we call the Philadelphia chromosome, right? And these two chromosomes shouldn't cross over. They have no business crossing over, but sometimes they do. Okay, and here you can see that Philadelphia chromosome and the cancer-causing gene got activated, all right? And the reason I say it's an interesting story, at least for you guys, you live right here, the East Coast of the United States. Oh boy, the scientific research that goes on all around where you guys live here in Bucks County, Pennsylvania. You can go up to New York or down to Baltimore and anywhere in between. And you can go further in either direction as well. The research is awesome. And the Philadelphia chromosome was first discovered and described from University of Pennsylvania, right? UPenn, School of Medicine. How cool is that? And also with work from Fox Chase Cancer Center Institute, right? So these things are both what? Less than 20 miles from where you live. And they make huge discoveries all the time and try to better us through really important healthcare. And again, a scientist from another amazing university, University of Chicago, helped understand the mechanism or identify the mechanism on how this non-homologous crossing over happened to cause this cancer gene from getting activated. All right, pretty crazy stuff, pretty awesome stuff that there's so many people out there working on this. So, comprende, how is reciprocal translocation different from crossing over? Again, you're crossing over between non-homologous chromosomes, right? Crossing over normally exchanges corresponding segments between homologous chromosomes. So that's pretty much the end of what we're going to talk about today. Hopefully you understand now why we reviewed meiosis and why it leads to such variation in a population, all because of the events that happen in prophase 1 and metaphase 1, and then how that random fertilization brings it all together, and wow, do we have a lot of variation in our population. And then we talked about, well, what happens if things go wrong, right? Nondisjunction, and it fails to come apart correctly. And a lot of times you can see that via a karyotype. Okay, but nondisjunction of autosomes, you usually don't hear about it much, probably because that developing offspring in there doesn't actually develop fully, right? Because it just, there's something going on there and it just stops developing, or there's a natural miscarriage of that. Or maybe it happened at a sex chromosome, and sex chromosomes, you get away with it a little bit more, right? So if you're a guy with an extra X, yeah, you're okay. You have Klinefelter syndrome, 47XXY, you're okay. If you're a girl that is missing an X or a Y, you're just 45XO, that's Turner syndrome. Okay, you know, there's some complications of both of these, by the way, but for the most part, you're okay, right? You might have to get checked every now and then. And then we talked about some chromosomal things that can happen as far as deletions or duplications or translocations and things like that. So not only gaining or losing chromosomes, but if the chromosomes themselves get goofed up, that can cause some problems. Okay, so Beaklings, that wraps up Unit 8, and I hope you found this video helpful. Okay, good luck.